Cancer is a horrible disease. Despite progress in some types of cancer, overall, cancer remains the second leading cause of death in the United States. It kills indiscriminately, taking old and young alike, and it affects everyone. I think there's no one that hasn't been touched by cancer, taking friends, family, or battling it themselves. Clearly, we need to do better. So I'm going to tell you today about some of the work in my lab based on a discovery we made about how the cell's shipping department plays a role in cancer and how we're using that to develop new therapies. This is definitely still work in progress, but gives you a peek under the hood about how my lab is approaching cancer. Before I do that, I'll need to provide some background. So, as most of you probably know, cancer is a disease of abnormal cell proliferation. So normally, most cells in our body do not proliferate. Instead, proliferation is orchestrated by growth factors that instruct cells when it's an appropriate time to proliferate. So examples include during the growth of tissues and organs during embryonic development, during growth from childhood to adulthood, and during wound healing. So these growth factors are small protein molecules that are released from some cells to regulate cell proliferation. Growth factor receptors act as receivers on the surface of cells to detect the presence of growth factors and consequently to trigger cell proliferation. When growth factors engage a growth factor receptor, the cells will proliferate. So cancer cells have many ways that they can circumvent the requirement for growth factors, including by producing their own growth factors, by having mutations in the growth factor receptor that keeps it stuck in the on state to incessantly drive proliferation, and by increasing the production of growth factor receptors so as to amplify their effect. Growth factor receptors are now validated targets for effective therapeutics in some types of cancer. So for example, in HER2-positive breast cancer, the drugs Herceptin and Perjeta act by blocking the HER2 growth factor receptor. Likewise, in EGFR-positive lung cancer, drugs like Iressa, Tarceva, and Tagrisso block the ability of the EGFR growth factor receptor to send proliferative signals. So that brings us to our research. So my lab has been doing research into the inner workings of cells with the idea that one of the limitations in our ability to treat cancer is because we have a limited understanding of how cells work. It seemed to me that if we understood better how cells work, we're likely to find new targets for therapeutics to treat cancer. One of the things we discovered is a key part of how cells move proteins from inside the cell to the surface of the cell, a process called secretion. So I'll show you a quick movie of a protein that we've labeled with green fluorescence, and we can watch it move from inside the cell to the surface of the cell. That process of secretion involves a part of the cell called the Golgi apparatus or Golgi complex, or more colloquially, the Golgi. The Golgi looks like a stack of pancakes and acts as the cell's shipping department. Protein molecules that we refer to as cargo enter the Golgi at one end of the stack, and then as they move along the stack, they are sorted, and at the other end of the stack, the cargo gets packaged into shipping containers that we call vesicles, where they are released from the Golgi for delivery to their destination, which for many, that destination is the surface of the cell. Among those cargoes are growth factors and growth factor receptors that go through the Golgi in order to be delivered to the surface of the cell. And their ability to function to drive cell proliferation absolutely depends on them being able to reach the surface of the cell. So we discovered the cellular machinery that packages cargo into vesicles involving a protein called Golf3. And it turns out that Golf3 and all of the proteins that we identified as being part of this process are all common drivers of cancer. At least 20% and maybe as many as 50% of cancers have activation of this Golf3 machinery. And it's important in many different kinds of cancers, including lung, breast, prostate, colorectal, pancreatic, and many other types of cancers. We also know that interfering with this Golf3 cargo packaging machinery shuts down growth factor receptor signaling and preferentially kills cancer cells. So that made us realize that if we could develop drugs that inhibit this Golf3 cellular machinery, that these drugs might serve as novel therapeutics for cancer. So we developed methods to identify chemical compounds that inhibit the Golf3 cargo packaging machinery. We screened 385,000 compounds and identified 15 inhibitors. And then I realized that, like most academic physician scientists, that I didn't really know how to go from chemical inhibitors to a viable drug. I realized that I needed help from experts, and for that, I applied for help from the Harrington Discovery Institute. Fortunately, I was selected for a Harrington Scholar Innovator Award. The Harrington Discovery Institute's mission is to accelerate promising discoveries into medicines for unmet needs. So that was exactly the help that I needed. The Scholar Innovator Program is Harrington's major diseases program. It's open to proposals for any type of disease, including many that focus on cancer, like my own. And that award came with a minimum of 2 years of support, which includes some monetary support, but more importantly, set me up with a team of experts with long and accomplished careers in the pharmaceutical industry who could provide advice on every aspect of drug development. So for me, I received advice on medicinal chemistry, which help us to develop improved chemical compounds that are more potent and have less toxicity. I also learned about how to think about actually giving a drug to a person, thinking about how to give it, and making sure that it would actually arrive at the cancer where we need it to act. So now, in our society, producing a new medicine for use in patients depends on pharmaceutical companies providing resources to fund the enormously expensive clinical trials that are needed to prove safety and efficacy of a new therapy. And those companies will only fund those trials if they see that there is financial incentive for them. And that depends on having solid protection of the intellectual property. So that's another thing that I learned about from my Harrington advisory team. So with the help of my Harrington team, we were able to develop improved compounds that effectively block Golf3 dependent secretion, block growth factor signaling, and kill cancer cells while being well tolerated by normal cells. Now, this is definitely still work in progress. Our compounds show promise and important proof of principle, but they're still not quite good enough to be medicines for use in people. But we're working on new approaches to make these better drugs. So now I thought I would take a few minutes to review my own career trajectory since I first received support from the Harrington Discovery Institute. At the time, I was a professor of medicine at the University of California in sunny San Diego, where my career was going great. But as I learned about what the Harrington Discovery Institute is doing, I realized the huge impact that their approach is likely to have on our ability to treat cancer and other diseases. And so I was able to convince my wife and daughter to leave San Diego and move to Cleveland so that I could join the Harrington Discovery Institute. Now, been and continue to be a pretty successful physician scientist. My lab made many important discoveries. We've published big papers in top journals. I've won awards, and I was well funded by government agencies and private foundations and served on many of their advisory boards. But it was clear to me that what Harrington Discovery Institute is doing is absolutely unique. Many funding agencies provide monetary support, but that is not sufficient to develop new medicines. Harrington Discovery Institute is unique in providing advice from experts on drug development, and that is key. So this is a very famous diagram of the hallmarks of cancer from Bob Weinberg and Doug Hanahan, which can also be thought of as a diagram of targets for cancer treatments. Now, my own lab's work targets proliferative signaling, but in just the last couple of years at the Harrington Discovery Institute, I've been able to steward programs that are developing treatments that target nearly every one of these areas. It's very exciting and inspiring. I'm convinced that the Harrington Discovery Institute's unique approach to team up academic physician scientists who have brilliant ideas about how to cure cancer with experts, to team them up with experts in drug development, and that this team approach is the way that we will cure cancer. So I'll thank you for your attention and thank you for your interest in thinking about how we can do better.